when pea plants that differ in two characters are crossed, what would happen? Mendel discovered the following 7 characters in pea plant: the seed shape, seed color, pod shape, pod color, flower color, flower location, and the plant height. So in seed shape, there is round and wrinkled as the opposite traits. Seed color, yellow, green. Pod shape, inflated, constricted. Pod color, green. Pod color, yellow. Flower color, purple, white. The location of flowers, axial or terminal. And plant height, tall pea plant or dwarf pea plant. So in each of these opposite traits, the one trait will be dominant. That is the one which is getting expressed in F1 generation. Like for example, in case of round and wrinkled seeds, regarding the character seed shape, round seeds are dominant and wrinkled are recessive. So whatever is located here in the top rows, they are all dominant traits, and the bottom rows, they are all recessive traits. For instance, when he crossed round green seeds with wrinkled yellow seeds, here round as well as wrinkled comes in characters seed shape, and green and yellow comes in the characters seed color. During gamete formation, the two genes which were present together, they will separate, and only one of the genes from each character will enter into gamete. So we get here capital R, small y, it is round and green. From that single gene only enters, not both the genes. And wrinkled yellow into the other gamete. Now both these gametes will cross, will fuse. After fusion, we get in the F1 generation, round yellow. That's because round seeds are dominant to yellow seeds. And now comes the self-pollination. So what all F1 plants are there that are round and yellow, they will be crossed among themselves, and that is called selfing. So we can see that now capital R gene, small r, capital Y, small y, they will independently arrange themselves and then they will fuse. The reason is because each of these genes, they present on separate chromosomes. The genes are not linked. They are present on separate chromosomes. Because of these reasons, they won't be aligned together. They will independently assort. And that goes for the Mendel's law of independent assortment. So while gamete formation, we get different combinations that can be written on one axis of Punnett square. So we got the following combinations of gametes from the first parent, and from the other parent also we will get the similar genotypes.
Now we have to cross between the gametes.
Now we get here round yellow. Second we will get round. This is also round yellow. This gamete is crossed with this gamete. In that way, the process continues. And then we have to go to the second row. So all these genotypes, whatever is there inside the Punnett square, they are the genotypes of the offsprings. So we get here round, yellow, round, yellow, because yellow is dominant, round, yellow. This is round, green, round, green, round, yellow. This is wrinkled yellow. So there comes a new combination. This new combination has come about because the genes were independently assorting. Now we'll write down the combinations. Round yellow. How many round yellow plants are there or genotypes are there? There are 9. The next combination is round green. Round 3. Then we'll start with the wrinkled. Wrinkled yellow. One here, second one here, third one here. This also be 3. And then comes wrinkled green. Wrinkled green, only one is there. So we get a dihybrid ratio. We call it dihybrid because it's a cross formed by crossing the plants that differ in 2 characters. That's why it's called a dihybrid cross. And the ratio obtained will then be called as dihybrid ratio. So we get here the dihybrid ratio -2 3:3:1. Now all the combinations in the progenies we obtained. Round yellow were the parental combinations. See, these were used as parents for crossing. Round yellow. But then after the cross, we are able to see new combinations. One is round green, and then we get the wrinkled yellow. These both are new combinations. Which we can call them as recombinants. Now we were able to get these new combinations different from the parents because during gamete formation, we can see that None of the two genes are placed together. So round and yellow genes and wrinkled and green genes, they all independently assorted. That is the reason why we got new combinations.